Okay, let's start. Topic, DNA and RNA. Okay, DNA and RNA class. What do you mean by DNA? It means deoxyribonucleic acid, and we have your RNA, which is ribonucleic acid. So for DNA and RNA class, we will be discussing the structure and the participation of DNA and RNA when it comes to DNA replication, DNA transcription, and translation. Basically, the gene expression. So ito po yung structure natin for the DNA and RNA. So let me give an example. This one is a double-stranded DNA or possible, yan, to. Yan. So helical, no? Yung helical structure na yan, class, kapag dalawa siya, double helix, kaya tinawag na double helical structure. Usually yung DNA, example for humans, no? Katulad natin humans, yung DNA po natin is double-stranded. And tinawag na double-stranded yan, it is because of this particular helix. Yung helix na yan, yung pag ganyan, na color blue na yan or purple ba yan, blue, it is made up of sugar, phosphate, backbone. And dyan po nakadikit yung base pairs. And backbone. So ito po, dyan po naka-incorporate yung ating DNA makeup, no, genetic makeup, yung mga nakita nyo nasa gitna. Yan. So sa kabila naman, ito example ng single-stranded natin. Yan. Okay. And then there's some base pairs, no? Or nitrogenous, or tawagin natin, sabihin natin nucleotide. Yan. Nucleotide or nitrogenous base. Okay. So either DNA or RNA, pwedeng single-stranded or double-stranded. Especially when we're talking about other organisms or such as viruses, no? May mga viruses tayo na double-strand, meron tayong mga viruses na single-strand. Example, COVID-19, di ba? COVID-19 is an example of an RNA. Yan. Okay? May mga viruses din tayo na DNA. Okay? Bacteria. So in general, yung tinutukoy ko dito, okay? When it comes to DNA and RNA. Pero mamaya, we will be focusing more on DNA and RNA in correlation with the human genome. Okay? So next. So ito maganda, no? We have here again the structure of the DNA. And on the other side, we have the structure of your RNA. Yung comparison lang ba, class, no? When it comes to the double, sa helical structure, itong sugar phosphate backbone, pareho lang sila. Pareho lang yan. Alam niyo kung ano pinagkaiba? Yung kanilang nucleotides. Yan. Example. For DNA class, we have the ATCG. Ano yung A? A stands for adenine, T for thymine, C for cytosine, and G for guanine. ATCG. And we follow a certain pairings. Kung makikita nyo, class, yung mga gitna, ito ha, yan. Yung mga yung mga base pairs sa gitna, dalawa yan. Kaya nga sinabing base pairs, no? Ganito yung tsura niyan, di ba? Sa gitna. Yan. Yung isa dyan, A, isa dyan, T. Okay? The other one is C, the other one is G. And we follow a certain format or structure. Si A, ang kapartner lang talaga niyan ay si T. Si C, ang kapartner lang talaga niyan ay si G. Hindi po pwedeng mag-cross. Example, sir, pwede bang si adenine ang ka-base pair niya ay si guanine? No. Si cytosine po ba, pwede ang ka-base pair niya si thymine? No. Okay? Yung buong DNA structure na yan, buong base pairs na yan, okay, AT at CG lang. Or pwedeng TA at GC. Yan lang yung pwedeng format na pwede or acceptable, no, for proper spacing. Yan, isa sa mga reason, proper spacing ng genes. How about your RNA? Ano napansin nyo sa RNA maliban sa single-stranded, no? Single helical. Yung kanyang nucleotide. Imbis na ATCG, AUCG. Why? Because say natin, A for adenine, C for cytosine, G for guanine. Anong naiba? Anong napalitan? Yung letter T, di ba? Nawala yung thymine class, no? It was replaced by uracil. Okay? Si uracil po ang pumalit. Ibig sabihin dito, class, once you see a base pair or genetic makeup, no, na ang laman ng base pair niya ay uracil, ibig sabihin nun, we are talking about RNA. RNA po yan. Wala pong DNA na ang kanyang nucleotide ay uracil. So, ang mangyayari dyan, class, na imbis na si adenine ang partner niya ay si thymine, mapapalitan yan pag RNA. Ang kapartner niyan si uracil. So, same pa rin, C and G. Cytosine 1 pa rin. So, pinagkaiba lang talaga ng DNA at RNA, itong presence of uracil for RNA. Kasi ang meron po sa DNA ay thymine. Take note of that, ha? That is a big difference. Okay, class? So, pasensya na kayo sa background noise. Okay, next. So, asan na ba yun? Next. So, difference between DNA and RNA. Katulad ng sinabi ko kanina, ito yung common na nitrogenous base nila. May adenine, may guanine, may cytosine, no? Meron din silang base, may sugar, may phosphate. Pero ang pinagkaiba is yung thymine for DNA and uracil for RNA. Another thing, yung kanilang sugar. Deoxyribose to, DNA natin, ribose lang for RNA. Class, malaki yung participation ng RNA when it comes to protein production. Okay? Explain ko mamaya. Next, let's move on now with the levels of chromosomal organization, starting with the chromosome. So, as we all know, kayo class, baka ang alam niyo sa chromosome, ganito yung tsura. Yan, diba? Parang rod shape yung mga arms, no? Rod. Actually, hindi. Yung mga ano yan, yung mga parts na yan, class, yung mga ang tawag dito, arms, no, ng chromosome natin. Pag zoom in ko yan, naka-coil yan. Tightly coiled. Yan, nakaikot siya. Ito yung centromere. Tapos yung arms, yan. Yan ang itsura ng chromosome talaga, class. Naka-coil. Okay? So, super coil within the chromosome. Kung isa-isahin natin yan, yung strand is made up of a chromatin, your chromatin fiber, and we have here your DNA. Yung DNA po natin, genetic makeup natin na yan, is hold, no? It is held by what we call histone. Okay? Meron pong nakadikit dyan, call it histone, which holds the DNA together with the nucleosome. Yung mga bilog-bilog na yan, that is nucleosome. Okay? Nucleosome, yung mga bilog-bilog. So, para mas maliwanag ito, mas detailed to class eh. Yung pareho lang actually yung ito tsaka to, mas detailed lang to, no, yung process. So, kung makikita nyo class, this is your DNA or genetic makeup, DNA. Okay? So, number 1, simplest level, we have your chromatin, yung strand. Number 2, complex with histones to form nucleosome. Ito yung ating nucleosome. Okay? So, ito yung histone class. Yung color, yellow ba yan or orange? Yellow, no? And then, yung yung nucleosome, itong purple. Yan. So, nakaikot po dyan and nakalock yung DNA strand natin. Okay? So, ayan. Diretso yan dito and eventually, it will undergo what we call folding. So, magkakaroon ng folds yan, class, katulad ng nangyari dito. And eventually, it will be tight under 300 nanometer fibers, compressed. Okay? So, magkakaroon ng folding yan na ganito, eventually, yan na. Ayun. Yan. So, naka-super coil yan dito. Yan. So, napakahaba po ng genetic makeup natin, yung DNA strand. Actually, mahaba po yan, no? Next, central dogma of molecular biology. Itong part ng discussion regarding DNA and RNA, ano to eh? It overlaps, no, with the chemistry or biochemistry. Kasi we will be talking about processes, no, structures. So, kung nakikita yung central dogma of molecular biology, yes, definitely, but it also overlaps with our discussion sa biochemistry. Ngayon, itong DNA replication, DNA transcription and translation. These are all what we call gene expression. Ibig sabihin, class, ito yung capability ng DNA natin. Okay, una, our DNA is capable of replication. Ibig sabihin, the same DNA structure can be replicated. Example, nasugatan ka, nadapa ka, nagkasugat ka. Di ba mag-heal yan? It will undergo healing. And it will regenerate. Cells will regenerate. When we say regeneration, class, it will develop, no, it will create another genetically the same cell as before. Okay, nagkasugat ka, syempre it will produce the same tissue, same skin cells. Okay, that is DNA replication. Yung skin po natin, continuous yung shed off niyan, 'di ba, skin cells? Nag-exfoliate yan eh, 'di ba, class? Continuous ang regeneration ng cell. Same, no? It undergoes DNA replication. So, isa na yon, DNA replication. So, ano naman itong DNA transcription? Ibig sabihin nito, DNA is converted into RNA, specifically mRNA. Okay? Sir, bakit importante na nagkakaroon din tayo ng conversion ng DNA into mRNA? It is because mRNA is important for the production of proteins. Just imagine, class, ha? Just imagine. Yung DNA natin not capable of transcription. Okay? Not capable. Not capable. So, ibig sabihin, walang mRNA. Di ba? Pag walang mRNA, walang translation into proteins. Class, di ba? Yung buong katawan natin is made up of protein. Am I correct? Di ba? Buong katawan natin gawa sa protein. So, hindi pa pwedeng walang translation. Kailangan talaga ng production ng protein. So, anong nangyayari? Yung DNA natin dictates to produce mRNA. And this mRNA now will undergo translation to produce specific types of proteins. So ano naman yung reverse transcription? Ito. Ano naman itong reverse transcription? Ganito, no? Ibig sabihin nito, from DNA to RNA and RNA to DNA. It can revert back to DNA. So one good example, class, nito, yung HIV virus, human immunodeficiency virus, HIV. Napakahirap, class, gamutin ng HIV up to na wala pang gamot dyan eh. Why? Because HIV is an example of a retrovirus. Tinawag natin siyang retrovirus kasi it can shift from DNA to RNA, RNA to DNA. Nagpapalit-palit po siya. Okay? Another example, class, yung test na PCR. Okay. Polymerase chain reaction. Actually, gawin nating RT-PCR. Yan. Di ba ginagamit to for COVID-19? Okay? Remember, class, si COVID-19 is an example of an RNA virus. Ngayon, ang problema, class, bakit ginagamit natin pang test yung RT-PCR for early detection? Ganito yon. Si PCR po can only detect DNA viruses. Okay? It can only detect DNA viruses. Paano ngayon si COVID-19 na detect? Ganito. Si COVID-19 RNA will undergo RT. Ano yung RT? Reverse transcriptase. Ibig sabihin, si RNA will be converted into DNA. Di ba? So, ganoon ang ginagawa, class. Iko-convert muna ni machine, okay, si COVID-19 RNA into DNA para mag-undergo siya ng PCR or polymerase chain reaction para mapadami yung virus. O, di ba? Example, si Sir Kabanatan, nagka-COVID, wag naman, no? Konti pa lang yung amount of COVID-19 ko sa katawan. Si PCR kasi pinapadami niya yon para ma-detect at the earliest time possible. Ang problema, ang PCR natin can only detect or duplicate viruses na DNA in nature. So, ito na, papasok na si RT, reverse transcriptase. Kino-convert na si RNA into DNA. Si machine ay nagpapadami sa loob ng machine. Okay? Ganun po. Okay, yung example ng reverse transcription. Okay, so now, let me proceed with the videos. Isa-isahin natin yan, class, no? Share ko lang. Okay, from time to time, magbabat in ako dyan, class, ha? Pag may mga kailangan tayong clarify or emphasize, okay? So, watch the video.
In this video, we will study about the nucleic acids. Nucleic acids are basically biopolymers which are made up of monomeric units known as nucleotides. We have two important functions for nucleic acids, that is, they hold our genetic information and they perform variety of another functions that we will discuss later. We have two important types of nucleic acids in our body, the DNA and the RNA. Let's first try to understand the definition of nucleic acids. So nucleic acids are biopolymers. Bio means that they are synthesized inside biological systems and they are not synthetically synthesized. They are polymers, which means they are made up of repeated units called as monomers, which in this case are nucleotides. Now take this example. We have many different types of biopolymers in our body. One of them are the proteins. And most of you know that the proteins are made up of repeated units called as amino acids. Similarly, nucleic acids are also important biopolymers in our body, which are made up of repeated units which are known as nucleotides. So the next important thing is to understand the basic structure of a nucleotide, which will help us to understand the structure of nucleic acids in detail. Now this is a simple diagram explaining the structure of a nucleotide. As you can see, the single nucleotide is made up of three important groups: the phosphate, the sugar, and the nitrogenous base. And these groups are attached to one another by bonds, which we'll discuss later. To understand the complete structure of a nucleotide, let's try to understand the structure of these groups one by one. So the first is the sugar. The sugar present inside the nucleic acids are 5 carbon sugars. Now most of you know the 6 carbon sugar present inside the body, and most important of them is the glucose. Now in chemical nomenclature, all the 6 carbon sugars are called as hexoses. The word hex meaning 6. There are different types of hexoses in our body other than glucose, like the mannose and galactose. Similarly, 5 carbon sugars are called pentoses, the word pent meaning 5. There are different types of pentoses in our body, but the two important types of pentoses that are present inside the nucleic acids are the ribose and the deoxyribose. Now this is a simplified structure of the sugar present inside the nucleotide. Now if we compare this structure with the chemical structure of the ribose and deoxyribose on the right, you can see the structure clearly. We have carbon atoms on each of these corners. Note that the last carbon atom is located outside the ring. We have an oxygen atom at the center topmost position. You can also see in this chemical structure that the carbon atoms are named from 1 to 5 in a clockwise direction. So the carbon atom next right to the oxygen is called C1, so C2, C3, C4 and C5. C5 is located outside the ring. You can also see in this chemical structure we have different type of functional groups attached to these carbon atoms, like the hydroxyl group and the hydrogen atom. The most important of these functional groups which determine whether the sugar is ribose or deoxyribose are present at the carbon number 2 and the carbon number 3. In the sugar ribose, we have a hydroxyl group that is attached to the carbon number 2. This is the chemical structure of ribose. Now if you compare this structure with the structure of deoxyribose, the only major difference is at carbon number 2, that the carbon number 2 contains only a hydrogen atom and not the hydroxyl group. So the resultant sugar is called a deoxyribose. Here you can see the carbon number 2 in deoxyribose contains only a hydrogen atom, whereas in ribose the carbon number 2 contains a hydroxyl group. You can also tell this by the difference in their names, that the deoxyribose has a prefix called as deoxy, which means removal of oxygen. The resultant sugar, the deoxyribose, is more stable as compared to ribose because of one less functional group. The deoxyribose is present in DNA and ribose.
Just looking at the structure, no, pinagkaiba lang nila dito for deoxyribose and ribose is actually the presence and absence of oxygen. Okay, sa ribose kasi andyan pa yung hydroxyl group natin, yung OH. Sa deoxyribose, remove na po yung oxygen. Actually, pareho lang lahat yan pag tinignan mo except for the presence and absence of oxygen for this particular deoxyribose and ribose.
Ribose is present in RNA. You can also remember this by the starting of the DNA, which starts with D, so the deoxyribose, and the RNA, which starts with R, so the sugar is the ribose.
Are you a student who learns from YouTube videos or attends online classes?
The next important group to study in the structure of nucleotide is the phosphate group, which is this entity right here. The phosphate group consists of a phosphorus atom in the center to which 4 oxygen atoms are attached, which are negatively charged. The phosphate group is the same phosphate group which is present in the adenosine triphosphate, the energy carrying molecule of the body. However, in ATP, we have 3 molecules of phosphate which are attached to one another by high energy phosphodiester bonds. The phosphate group is a polar molecule due to the presence of highly ionized oxygen atoms which impart negative charge to the phosphate group. Now, the third important structure present in the structure of a nucleotide is the nitrogenous base. Nitrogenous bases are basically molecules that contain nitrogen in varying amounts and they act as a base. Now, most of you know that the human body contains organic chemicals which have carbon, hydrogen, and oxygen in varying amounts. In nitrogenous bases, nitrogen combines with all of these atoms to form ring-like structures. These molecules are called bases since they can donate electrons to other molecules and form new molecules in this process. The nitrogen combines with all of these other atoms to form ringed structures. Now we can have two different types of ringed structures. One where there are single rings and second where there are double rings. The single rings are called pyrimidines and the double rings are called purines.
Okay, class. So here, we will differentiate yung type ng nucleotide based on the type of rings na meron siya. So pag pyrimidine, single ring. Kapag purines, double ring. So what are those nucleotides? Ito yung adenine, thymine, cytosine, guanine, uracil. Remember class si uracil can only be present on RNA, ribonucleic acid. Hindi po siya present sa DNA. So check natin kung saan bang magpo-fall yung structure ng different types of nucleotides. Is it pyrimidine or purine?
We have 3 different types of pyrimidines, the thymine, cytosine, and the uracil. And we have 2 different types of purines called adenine and guanine. In the center, you can see the chemical structure of each of these bases. As you can see, the purines, which are double ring structures, adenine and guanine. Due to the presence of double rings, these purines are larger as compared to the pyrimidines. The thymine, cytosine and uracil all are single ring structures and they are smaller as compared to the purines. The difference in the size of these bases is very important as it helps to pair these bases in the structure of DNA properly, which we'll study later. So now you know the individual structures which are present in a single nucleotide. Next we will look at how these individual structures bond to each other to form a single nucleotide. And then we will look at how many nucleotides bind to each other to form a chain of nucleotides, which is called a polynucleotide, which can be either DNA or the RNA.
Dito class, you can see ito yung one part of the DNA strand. Sabi natin kanina, yung DNA natin pwedeng double helix and dalawa yung nucleotides na magkadikit. Dito class, pinapakita yung isang strand ng DNA or pwedeng RNA. Okay, kasi at this point single strand yung nakalagay Hindi naman binanggit kung DNA or RNA yan. Nakalagay nga dyan, either it is DNA or RNA. But definitely, ito ay isang part ng nucleic acid. Kitang-kita naman dyan, yung color pink is the sugar phosphate. We have the sugar and yung color green po would be the nucleotide. Yung green na yan, it could either be adenine, guanine, thymine, and so on. So let's proceed.
So if we take a sugar and name just the important carbons here, which will be the carbon number 1 and the carbon number 5, and of course you can see that at the carbon number 2 there is only a single hydrogen atom, so this sugar will be a deoxyribose. Now to pair this deoxyribose sugar with this pyrimidine ring, the bond formed will be between the N1 of the pyrimidine ring and the C1 of the sugar. If you want to go into the details about how these nitrogens are named, you can check the link in the description below for the IUPAC nomenclature of the aromatic compounds. Now this bond will be an example of a glycosidic bond, which is a covalent bond which links carbohydrates with other structures. Now if we have to pair a purine ring at a similar position, the bond formed will be between the N9 of the purine ring. Now next, let's attach the phosphate. The phosphate group attaches with the carbon number 5 of the sugar, and the bond formed is the example of ester bond, which is also a strong covalent bond. So now you know how the three structures in a single nucleotide bind to each other to form one nucleotide. Next, if we look at how many nucleotides bind to each other to form a chain of nucleotides called polynucleotides. So you can see basically what happens is that the phosphate group that attaches to the carbon number 5 of the sugar below also forms a bond with the carbon number 3 of the sugar above. And this pattern is repeated again and again. So you can see that the phosphate groups forms bonds both above and below the chain of these nucleotides. So as a result, a long chain of nucleotides is formed, which consists of a sugar phosphate backbone, and in the center we have all these nitrogenous bases which project from the sugar phosphate backbone. Now this can be an example of RNA if the sugar present in this example will be a ribose sugar. But if we take a similar strand of polynucleotide and run it in opposite direction, and this strand binds with the original strand through hydrogen bonds. Now here comes the role of complementary base pairing. Now what complementary base pairing says that only adenine forms a hydrogen bond with thymine and only guanine forms a hydrogen bond with cytosine. Now this is the universal rule that is applied in the structure of a DNA. So here you can see adenine forms a hydrogen bond with thymine, and similarly thymine forms
Okay, as you can see, meron na tayo ditong nabubuo no, na sabihin na natin DNA, no? DNA strand, double helix. Gusto ko lang i-emphasize dito yung position ng helix, yung sugar phosphate. And actually yung buong structure ng nucleotides, no? They are in the opposite direction. Yung isang side pataas, yung isang side kabilang side pababa. Again, yung nasa may left side pataas, yung nasa may right side pababa, no? Yung binding nila is in opposite direction. It is very important kasi mamaya babanggitin yan during replication, transcription, and translation. Let's continue.
hydrogen bond with only adenine. In similar case, guanine forms a hydrogen bond with cytosine and vice versa. Now this is the basic structure of DNA, and you can compare this with the step ladder structure of the DNA. You can see that the supports of the ladder in the red are basically representing the sugar phosphate backbone, and the rungs of the ladder represent the nitrogenous bases that are forming hydrogen bonds with each other. Now of course the original structure of DNA is that of a double helix. So you can compare this structure with a double helix where the helices of this structure.
Dito class, meron siya tinatawag na ladder type, no? Actually, this is the double helix structure. Kaya lang, in-straighten niya para mas makita natin yung base pairs sa gitna. Example, di ba? GC, AT, TA, CG. So, mas kita natin yung structure. So, kung tatanungin niyo kung ano to, this one is a DNA strand. Kasi walang uracil, no? Pwede mo lang sabihin na ito ay DNA strand. Again, wala pong uracil, no? Yung adenine natin is combined with thymine. So this is definitely a double-stranded DNA. Continue natin.
represent the sugar phosphate backbone, and in the center you can see the red lines which represent the different nitrogenous bases that are forming bonds with each other. So this was an introductory video explaining you the structure of nucleic acids. In subsequent videos, we will study in detail the structure of DNA, the structure of RNA, DNA replication, transcription, translation, and many other topics. So make sure to subscribe to our channel for further upcoming videos. In this video, we will study about the DNA replication. DNA replication
And
is the
next, DNA
biological process
replication.
of producing two identical replicas of the DNA from one original DNA molecule. And as simple as it may seem, it is one of the most complicated biological processes and involves the accurate copying of almost 6.5 billion base pairs in a complete set of DNA during each cell division. DNA replication takes place inside the nucleus of the cell, where two identical and new copies of the DNA are synthesized from the original DNA. The newly formed set of DNA will then be transferred to the future nucleus that is produced during the process of cell division. At the end of cell division, we have two completely new cells, each having its own set of DNA. Now, in the previous videos in this section, we discussed in detail about the structure and function of the DNA, about nucleic acids, and we also talked about the concepts of complementary base pairing and directionality. These concepts are essential for understanding the process of DNA replication, so make sure to watch both these videos for a better understanding of this section. Now let's talk about the overview of the process of DNA replication. What you see here is the simple double helix model of the DNA. In this model, you can note that it consists of these two blue lines which basically represent the sugar phosphate backbones of the DNA, and at the center we have these orange lines which represent the nitrogenous base pairs of the DNA. If we untwist this double helix model, we will get something which is known as the step-ladder model of the DNA, which is a simple depiction of the structure of the DNA.
Ngayon ko lang nakita yung inside ng engine ko.
I was surprised with how clean it is.
So in the long run, na-maintain mo na maayos yung makina mo.
Use good quality fuel such as Caltex. It really boosts performance.
In this example, you can see that the DNA consists of these two identical strands which almost run parallel to each other. Now, in the process of DNA replication, what happens is that these two strands separate.
Okay, class, ito na yung highlight ng DNA replication. So we have here a two identical structure of DNA, the left and the right, di ba? Sa ladder type natin, mas makikita natin yan. Then, ang mangyayari dito, class, there will be a separation of these two DNA strands, no? The left and the right. Bakit? Gagawin natin template yung bawat isang strand. Okay? So, ibig sabihin, there will be a formation of another DNA on the other side of each of those template. So, pag hinati ko sa gitna yan, kulang na, di ba, class? So, ang papalit dun sa kulang ay another same, no? Structure, same genetic makeup nung DNA strand natin na template. So, ang magiging end product dito, class, from one double helix DNA strand becoming a two double helix DNA strand. Identical. Okay? So, ganoon din yung mangyayari. So, meron ka ng dalawa. Same. Yung dalawang yun, mahahati sa gitna. Okay? Magkakaroon ng template. Mayroong papalit na bagong strand, which is identical. So, magkakaroon ka ng 4. Ulit-ulit yun. Paulit-ulit. Parami ng parami. Ganon siya dumadami, class. Ganon siya nagre-replicate. Nahati sa gitna, napapalitan ng bago, okay, and then it will undergo again replication. So tingnan natin.
Original DNA which separates to form these two strands is called the parent DNA, and then these two strands which are separated from each other act as templates upon which new strands are copied and formed. Now, this process is made possible by the concept of complementary base pairing. Basically, we discussed the process of complementary base pairing in detail in the previous videos, and in a nutshell, you can note that the DNA consists of 4 nucleobases, which are the adenine, cytosine, guanine, and thymine. In the structure of the DNA, the base adenine always pairs with the base thymine and the base guanine always pairs with the base cytosine. This is called the Chargaff's rule of DNA complementary base pairing. This is done so to maintain a proper distance between the two strands and also maintains a proper stable structure of the DNA helix. When the parent DNA is split, each strand acts as a template for the formation of new strand. This leads to formation of two completely new sets of DNA which are called as the daughter DNA. Now if you take a closer look at this step ladder model of the DNA, you can note that it consists of these two strands which are joined together in the center by hydrogen bonds. Now these two strands run in completely opposite direction to each other, which means they are anti-parallel, and they also have a direction. Now in DNA we don't have the north, south, east or west directions, but on DNA we have two ends, a 5' end and a 3' end of this strand. And since the strand above is running in completely opposite direction, it will have its 3' end on this side and its 5' end on this side. So due to this, the strand below is said to be running from the 5' to 3' direction, and the strand above is
Class, I would like to emphasize that the creation of the new DNA strand will be starting at the 3' end. Kaya very important dito yung direction of the DNA strand. Again, yung strand po natin, new strand of DNA will start at the 3' end.
said to be running from the 3' to 5' direction. Now to understand the concept of direction in DNA in a better way, let's take a brief and close look at the structure of the DNA. Now in this example, you can see the different nucleotides and how they are joined together in one strand of DNA. If we take one nucleotide out from this strand, you can see the simple depiction of the chemical structure of a nucleotide. We will not go into the details as we have discussed the structure in detail in previous videos, but you can note that we have three important groups in a nucleotide, which are the phosphate, the sugar and the nitrogenous base. Now if we isolate the deoxyribose sugar from this nucleotide, we can see that the carbon atoms in the deoxyribose sugar are numbered from 1 to 5 in a clockwise manner with the fifth carbon outside the ring. Now you can compare this illustration with the chemical structure of deoxyribose for a better clarification. And it is the fifth carbon to which the phosphate group is always attached. Now if we take a look at the structural illustration of a single strand of DNA again, you can note how the deoxyribose sugars are arranged in this single strand, and you can also see how all the deoxyribose sugars are aligned in this direction. Due to this orientation, the fifth carbon is always located on this side to which the phosphate groups are attached, and the third carbon are always located on this side to which the hydroxyl group is attached. This defines the 5' end and the 3' end of this strand of the DNA. Now if you take a look at the second strand of the DNA, you can note the key difference, that is that it runs in completely opposite direction, which you can clearly tell by looking at the orientation of the sugar molecules in this strand. These sugar molecules are aligned in this direction, which is completely opposite to the strand below. And this defines the 3' end and the 5' end of this strand. Now the question arises, why is the concept of direction important in DNA replication? Well, this is because the main enzyme of the DNA replication, which is DNA polymerase, it can only add new nucleotides as a 3' end of the DNA. This means that the process of DNA replication can only progress from the 5' end to the 3' end of the DNA. This is because the new nucleotides can only be added at the 3' end. Well, we will get into the details of the concept of direction in DNA replication later in this video. Now let's talk about the various enzymes that make this complex process of DNA replication possible. In this table, we will study about the name, the symbol, and the function of the enzyme involved in the process of DNA replication. The first enzyme that we will discuss is the enzyme helicase, which will be denoted by this symbol. Now, the main job of the enzyme helicase is that it unzips the double helix structure of the DNA. The enzyme helicase performs this function by breaking the hydrogen bonds between the base pairs. This separates these two strands, creating the replication fork upon which various other enzymes will act in the process of DNA replication.
Woo! Adapted mind math! Come explore the site with me.
So the next enzyme we will discuss is the enzyme DNA polymerase, which will be denoted by this symbol. The DNA polymerase performs the vital function of formation of a new strand of DNA known as the daughter strand. Now in this example, you can see the replication fork created by the enzyme helicase. And here is the 5' end on the left and the 3' end on the right of the upper part of the DNA. The DNA polymerase performs the function of formation of a new strand by arranging new nucleotides in the form of a.
Okay, class. Kung mapapansin nyo, after the helicase, ang purpose ng helicase natin, di ba, is to unzip the DNA strand, no? So, yung base pairs kanina, hindi na sila pairs kasi hinati na natin sa gitna. Ang purpose nun, for us to have a template. Ngayon, kapag may template na tayo, new nucleotides will attach to those new templates, di ba? Tulad niyan, yung may mga letter T, those are new nucleotides that will attach to the template, creating a new double helix, no, double-stranded DNA. So, pero syempre, it will follow pa rin yung ATCG, adenine thymine, tsaka cytosine guanine na tawag dito, combination. So, let's proceed.
DNA strand by using the parent DNA as a template. Now you can also note that the DNA polymerase cannot synthesize a brand new strand but requires a small sequence of RNA to do so. This action is performed by another enzyme. So the next enzyme which we will discuss is the enzyme RNA primase. The RNA primase will be denoted by this symbol. Now, like we just discussed, the DNA polymerase cannot initiate the synthesis of a new strand of DNA from scratch. So the enzyme primase synthesizes a short segment of RNA known as the primer, which acts as a guide for the DNA polymerase to synthesize the strand further. Now this short segment of primer is later removed and replaced by a new segment of DNA. The next enzyme we will talk about is the DNA ligase, which will be represented by this symbol in the process. The DNA ligase performs a vital function of joining together of the Okazaki fragments on the lacking strand. Now this may seem complex right now, but we will get to this in detail later in this video. And the final enzyme we will talk about is the enzyme topoisomerase, which will be denoted by this symbol. Now the DNA is a double helical structure which consists of two strands of DNA which are twisted around each other. When the enzyme helicase opens up the DNA, there is generation of stress in DNA strands which can lead to supercoiling of DNA. Now this process is solved by topoisomerase, which cut and reseal the fragments of DNA to relieve the stress that is produced by unzipping of the DNA.
Ko lang pala i-emphasize din class, no? I think mali-discuss din naman later sa video na napakahaba ng DNA strand natin, di ba? Sabi ko nga naka-coil, no? Sobrang haba niyan. Ang tanong, yung example, yung helicase ba, it will unzip from tip to tip nung DNA strand natin, genetic makeup, dulo-dulo, as in sobrang haba nun, di ba? No, no? May mga specific portion lang ng DNA class na kinakat, no? And then, yun po yung kinakopy. Yun yung dinuduplicate or nire-replicate. So, maraming portion ng DNA, genetic makeup natin, DNA natin, yung kinakat and nire-replicate by portion, hindi po isang bagsakan. Okay? So, hindi yung buong napakahabang coil ng DNA mo ay ina-unzip at lahat yun sabay-sabay nilalagyan ng new nucleotide, new template, no? Lahat hindi. May mga specific portion ng DNA natin na nag-undergo ng replication by portion. Okay? So, continue natin.
So this was part 1 of this video in which we discussed the overview, the concepts of directionality, and the various replication enzymes involved in the process of DNA replication. In part 2 of this video, we will study about the actual replication process. Thank you so much guys for watching this. In this video, we will study about the process of the DNA replication.
Part 2, DNA replication.
In part 1 of this video, we discussed in detail about the overview of DNA replication, the concept of directionality of DNA in DNA replication, and also about the various replication enzymes that are involved in the process. Now in part 2 of this video, we will study in detail about the process of DNA replication. Now, for the process of DNA replication to begin, it is important for the two strands of the DNA to open up. This job is done by the enzyme helicase, which is also known as the unzipping enzyme. The enzyme helicase performs this function by acting on the hydrogen bonds which are present between the base pairs of the opposite DNA strands. The helicase breaks these hydrogen bonds, due to which the double helix opens up, leading to the creation of the structure known as replication fork. This exposes a sequence of DNA base pairs upon which many enzymes will subsequently act. Now, the process of unzipping of DNA involves action of multiple helicase enzymes on different segments of DNA. These segments of DNA where the enzyme helicase acts are also known as the sites of origin.
Yung sinasabi ko kanina, class, no, na yung DNA replication by portion. So, ang daming helicase dyan, di ba? Na nakapatong sa taas. Ibig sabihin, there are portions of the DNA that will undergo replication, okay, by portion, hindi isang helicase lang for the whole genetic makeup or DNA, okay?
Now, since the helicase acts on the DNA at multiple sites, multiple replication forks are created upon which the process of DNA replication will proceed. Now, to prevent the two separated strands of DNA from joining together, a special type of protein known as the single-stranded binding protein holds the two strands of the DNA apart. According to the concepts of directionality we learned in part 1 of this video, let's suppose this is the 3' end of the upper strand and this is the 5' end of the upper strand, and similarly we have the 3' end of the lower strand in this direction and 5' end in this direction. Now, the main enzyme, the DNA polymerase, which has the job of formation of new strands of DNA, can only form a strand from the 5' to the 3' direction. Now, since the upper daughter strand of this DNA is running from the 3' to 5' direction, the DNA polymerase can easily form the replicated opposite strand from the 5' to 3' direction. And since the helicase is also unzipping the DNA in the same direction, the DNA polymerase can form the new strand continuously. But when we look at the opposite strand of DNA which is running in the opposite direction, things are very different. Since the DNA polymerase has to act, it has to form the new strand in this direction, which is in the 5' to 3' direction. But since the helicase is opening the DNA helix in the opposite direction, the DNA polymerase cannot synthesize the new strand continuously. Well, this problem is solved by formation of the new strand of DNA in short segments known as the Okazaki fragments. These fragments are later joined by a different enzyme to produce one continuous strand. Due to this reason, the opposite strand is known as the lagging strand, and the replication on the lagging strand is discontinuous replication as compared to the continuous replication on the leading strand. Now we will take the leading and the lagging strand separately and understand the process of DNA replication further. So after the formation of the replication fork, the next important step is the process of formation of RNA primer. Now what happens is that the DNA polymerase enzyme can only add new nucleotides to the 3' end of the existing nucleotide strand, which basically means that the DNA polymerase cannot start synthesizing new DNA out of nowhere and basically requires a short segment of nucleotides so that it can add new nucleotides as a 3' end of this sequence to form the new DNA. This problem is solved by the enzyme RNA primase. The primase makes an RNA primer, which is a short stretch of nucleic acids complementary to the template that provides the 3' end for the DNA polymerase to work on. So now we have a leading strand of DNA which is running from the 3' to the 5' direction, and we already have the short primer synthesized by the enzyme primase. Now it is the turn of the enzyme DNA polymerase to do its job, and a few important points that you should know about DNA polymerase are that it always requires a primer to work with, which is a short sequence of nucleotides, and it cannot start synthesizing the new DNA from scratch. The DNA polymerase can only add new nucleotides at the 3' end of the DNA and can only synthesize the new DNA in one direction. And also the DNA polymerase requires energy for polymerization reactions that it gets from the nucleotides itself. Now once the primer is synthesized, the DNA polymerase can perform the vital function of elongation of DNA strand by addition of new nucleotides to the 3' end of the growing chain. The DNA polymerase uses the deoxyribonucleotides, which contain either of the four bases, the adenine, cytosine, guanine, or thymine. The DNA polymerase adds these nucleotides one by one to the 3' end of the new DNA strand. Now, since the enzyme helicase is opening the DNA helix in the same direction, the DNA polymerase can synthesize a long chain of new DNA before separating, and this is known as the continuous synthesis on the leading strand. The DNA polymerase also requires energy for this polymerization reaction, which it basically gets from breaking the phosphate bonds present between the triphosphates of the deoxyribonucleotides. These phosphate bonds are high energy bonds which, when broken, provide the energy for this polymerization reaction. Now we will take a look at the lagging strand, which runs from the 5' to the 3' direction. We can see that the main problem on this strand is that the DNA helix is being opened up in the opposite direction of the direction in which the DNA polymerase synthesizes the new DNA, which is from the 5' to the 3' direction. Now this problem is solved by formation of new DNA in short segments known as the Okazaki fragments. What happens is that on the lagging strand, the primase synthesizes a short piece of primer, and then the DNA polymerase synthesizes a short segment of DNA and then separates from the growing end. After this, the enzyme helicase would have opened up more of the DNA helix in this direction. Then again, the enzyme primase synthesizes a new primer further up on the DNA.
Okay, emphasize ko lang ulit class, no? Remember, sinabi ko kanina na new strand of DNA nucleotides can only be attached on the 3' to 5' end. Kapag 5' to 3' end, hindi po pwede. No? Kaya ang ginawa dito class sa 5' to 3' end, may kita nyo naman nasa baba, no? 5' to 3' end, kailangan mo ng primer. Di ba? Why? Because they only made a strand or tinatawag na fragments of DNA strand, hindi buo. So dun sa mga spaces na blanco or walang nucleotides, naglagay tayo ng primer. So temporary lang yung primer niyan kasi mamaya matatanggal din yan and it will be replaced by a true nucleotide. Okay?
Which is followed by formation of another short segment of DNA. This process is repeated multiple times, thus known as the discontinuous DNA synthesis. Now, after the DNA polymerase has done its job, the newly synthesized DNA looks something like this. It has short segments of DNA with interrupting segments consisting of RNA primer. After this, another enzyme known as the exonuclease comes into play, which removes these short segments of primers, which are then replaced by short new segments of DNA by another type of DNA polymerase. After this, the enzyme DNA ligase seals or joins the DNA fragments to produce a continuous DNA strand even on the lagging strand. Lastly, it is also important to check for errors in the newly synthesized DNA, and this is done by another type of DNA polymerase, which proofreads the newly synthesized DNA for any wrong bases that may have been inserted, and if it finds a wrong base or sequence, it removes the base and then replaces it with a correct one. After the process of DNA replication is completed, the two newly synthesized DNA molecules coil up again into the DNA helix model. So this was a brief overview about the process of DNA replication. Guys, thank you so much for watching this video. If you want to see the genetic
Next, we
material
have
is stored in the form of
transcription.
DNA in most organisms. In humans, the nucleus of each cell contains over 3 × 10⁹ base pairs of DNA, distributed over 23 pairs of chromosomes. Each cell has 2 copies of this genetic material. This collectively is known as the human genome. The human genome contains about 30,000 genes, each of which codes for certain proteins. Although thousands of genes in the human DNA code for proteins, large stretches of DNA in the human genome are transcribed, but they do not code for proteins. These regions are known as introns, and they make up about 95% of the genome.
Okay, class. Now, ito, explain lang natin, no? Meron tayong tinatawag na introns and exons, no? And introns does not code for proteins. These are part of the genes na hindi nagko-code ng protein. So actually class, hindi natin kailangan to. Okay? Alam nyo class, ang end product ng transcription natin, di ba, is RNA. To be more specific, mRNA, messenger RNA. Ang laman lang dapat ng messenger RNA natin or mRNA is exons. Why? Yung exons kasi class, they code for proteins. Yun yung kailangan natin in order for this mRNA, no, to proceed with translation. Di ba? Anong end product ng translation? Protein. Just imagine class, paano kung ang laman pala ng mRNA natin puro introns? Does it code for proteins? Hindi. Di ba? So, useless. So, definitely class, sa end ng bandang end part nitong discussion, no, doon yung makikita na if in case may nakasingit na introns, okay, tinatanggal sila during the process. Kasi dapat exons lang po laman yan in order for it to code for proteins. So, tuloy natin.
Some of this non-coding DNA controls gene expression, but the purpose of much of it is not yet understood. This is a fascinating subject that is certain to advance rapidly over the next few years. So we know that all the information of the human genome is coded in the DNA in genes in the form of nucleotide base pairs. How this coded information is used to produce functional products like proteins is known as gene expression, and it essentially consists of two important steps. The first involves copying of the coded information in DNA to a RNA molecule, which takes place inside the nucleus of the cell, and this is known as transcription. This RNA molecule then leaves the nucleus, and the information of this RNA molecule is used to produce functional gene products like proteins, and this step is known as translation. In this video, we will take a brief look on the process of transcription. Make sure to subscribe for upcoming video on translation. You can also take a look at our previous videos on DNA structure and DNA replication. Links are in the description below. So the process of transcription takes place in two broad steps. First, the enzyme RNA polymerase forms a pre-messenger RNA molecule. This happens in the nucleus by using base pairs complementary to the base sequence of the DNA template strand. In the second part, the pre-mRNA is edited by more enzymes to produce a desired mRNA, which is the final product of the transcription. So let's look at the first section of this process, which is the formation of the pre-messenger RNA, which essentially takes place in three steps: initiation, elongation, and termination. Now the main enzyme that is involved in this process of transcription is the RNA polymerase. The RNA polymerase has the main job of copying the information of the DNA to a new RNA molecule. RNA polymerase builds the new strand from the 5' to the 3' direction, adding new nucleotides to the 3' end. So the first step in the formation of a pre-messenger RNA molecule is the initiation, in which the enzyme RNA polymerase binds to the promoter region of the DNA. Now let's suppose this is a gene that is shown in the green that needs transcription. At the beginning of each gene, there is a sequence of base pairs known as the promoter region of the gene. The promoter region serves as a guide which tells the enzyme RNA polymerase where to attach. Once the RNA polymerase binds to the promoter region, it separates these two strands of DNA, providing the single-stranded.
Kung mapapansin nyo class, no, parang same lang, no? Nagkaroon din ng unzipping, okay? Yung promoter region, ibig sabihin, ito yung certain part or specific part ng DNA na gagawing template in order to create an RNA. Okay? So, ibig sabihin, sabi nga natin, no, conversion of DNA to RNA. Ibig sabihin, yung DNA natin mahati sa gitna to serve as a template, and yung idadagdag dyan na nucleotide, no, na imbis na A, T, C, G, mapapalitan ng A, U, C, G, which is uracil. Okay? So, ganun po ang mangyayari dyan. Okay, tingnan natin. Tuloy natin.
Template needed for transcription. Only one of these strands is used in the process of transcription, which is known as a template strand. This strand always runs from the 3' to the 5' direction. This is because of the fact that we just discussed. That is, that RNA polymerase can only synthesize the new strand from the 5' to 3' direction. So it can only use a strand of the DNA running from the 3' to 5' direction as a template, as the new strand that is copied is always copied in the anti-parallel direction. So after
Class, pansinin niyo yung yung new strand no na kumabit doon sa may DNA natin. Yung kulay purple. Merong diyan, U, no? May uracil na kasi ang na-create na natin diyan na strand is actually RNA na.
the RNA polymerase binds to the promoter region of the DNA, the second step in this process is elongation, which is the process of copying the bases on the DNA template strand to form the new complementary RNA strand. The RNA polymerase uses nucleotides according to the base pair complementary rules to create a RNA copy. In the previous videos, we learned how the DNA is copied by DNA polymerase to form an identical new strand of DNA. In that case, the DNA polymerase added the base thymine when the template strand had the base adenine, and it added the base cytosine when the template strand showed the base guanine, and vice versa. But in case of transcription, the RNA molecule that is produced contains the base uracil instead of thymine. So there are no T's in the base pair sequence of RNA. Whenever the RNA polymerase detects the base adenine in the template strand, it adds uracil in the forming RNA strand. So this is how the enzyme RNA polymerase adds new base pairs in the 3' end of the growing RNA chain, and this is known as elongation. The next step is termination. So just like we had a sequence of gene known as the promoter region which signaled the enzyme where to start, we have regions of the gene that basically signal the enzyme where to stop. These signals are known as terminators, and they signal that the RNA transcript is now complete. Once the RNA polymerase reads and transcribes the terminator regions, the newly formed RNA transcript is released from the transcription complex. So this was the first section of this process in which the DNA is used to produce a RNA molecule. This RNA is known as the pre-mRNA or the pre-messenger RNA. The prefix is used here to show that this mRNA is not yet ready to leave the nucleus and start the process of translation. So the next section of this process of transcription deals with how this pre-mRNA is converted to a mature RNA molecule. Imagine that you want to publish a new book and you have printed all the pages. Now you cannot sell the book yet because the new book needs covers, both front and back. Also, before selling it, it is important to check for any blank or wrong pages that may have been inserted. These two processes essentially represent how the pre-mRNA is processed. The first step involves addition of a cap and a tail to the ends of this pre-messenger RNA molecule. These caps and tails are short amino acid sequences that protect the pre-mRNA molecule. The free ends of the pre-mRNA are liable to attack from many enzymes. The cap and the tail covers these ends. The second step of this process involves removal of any unuseful or junk sequences that do not code for proteins, and these are known as introns. This is known as
Okay, class. Ito yung mention ko kanina na yung mga junk sequences, which is introns, which does not code for proteins, tinatanggal siya dun sa ating pre-mRNA. And bakit merong cap and tail? Ang purpose ng cap and tail is for protection so that it will not alter the RNA. Example, certain enzymes can alter the RNA. So pinoprotektahan niya yung RNA natin dito. So after the cap and tail and removal of these junk sequences, saka pa lang ito magpo-proceed into mRNA. Okay? Kapag mRNA na yan, class, ready na yan to go to what we call endoplasmic reticulum ng cells. Alam niyan yung ER, di ba? So specifically, yung tinatawag natin na rough endoplasmic reticulum. Bakit? Si mRNA po kasi, pumupunta yan sa ribosome. And as we all know, ribosome encourage nor promotes the production of proteins. So tuloy natin to.
RNA splicing, in which the protein RNA complex known as the spliceosome removes these non-coding parts. The rest of the DNA is then joined again together to create a mature RNA molecule. So this was a brief look at the process of transcription. Guys, thank you so much for watching this video. If you found this video and all the other free educational videos This video is made possible by Marrow. Marrow is the
Last
new
video, class,
revolutionary
we have
platform
DNA
for NEET PG, AIMS,
translation.
PGI, and DNB exams.
mRNA
With over
produced
640 hours of recorded video
last
lectures and more than 28,000 highly selected MCQs on all undergraduate medical subjects, Marrow focuses on concept-based learning, which is essential to perform better in the PG entrance exams. Marrow has chemical reactions used in the synthesis with image-based question description below. In this video, we will learn about the process of translation. Translation is the second step in gene expression. Gene expression is the process by which information stored in your DNA in the form of genes is used in the synthesis of functional gene products. These gene products are mainly proteins which have numerous functions in your cells like repair and maintenance of cells, energy synthesis, and enzymatic actions on biochemical reactions. Gene expression essentially occurs in two steps. It begins in the nucleus with the process of transcription, in which the information on the DNA is copied into a RNA. This RNA is known as the messenger RNA or the mRNA, since the main job of this RNA is to carry this information or message outside the nucleus and use it to perform the second step of gene expression known as translation. In translation, the message stored on the mRNA is decoded in a ribosome to produce a specific amino acid chain or polypeptide. Make sure to watch the video on the first step of gene expression, the transcription, for a better understanding of the complete topic. Also make sure to watch the videos on structure of nucleic acids and DNA, as it will help you to utilize this video in a much better way. All the links are in the description below. Let's first have a brief look onto what are the structures in the cell that make the process of translation possible. Besides the messenger RNA, translation also requires another type of RNA known as the transfer RNA or the tRNA. Ribosomes are dedicated cellular machineries that make this whole process possible. The ribosomes read the message on the mRNA, and the tRNA transfers individual amino acids to the ribosome according to the sequence of base pairs on the mRNA. These amino acids are then joined together by bonds to form a protein. So the process of translation occurs in three basic steps. First step is known as initiation, in which the ribosome assembles around the target mRNA. In the second step, known as elongation, the tRNA transfers amino acids to the ribosome, which are joined together to form a polypeptide chain. In the last step, known as termination, the ribosomes release the polypeptide when it reads a stop signal on the mRNA. Don't worry if you don't understand these steps yet. We will look at the details of this process in just a bit that will help you to understand this complex cellular process. Now let's look at where this process takes place in the cell. It's slightly different for eukaryotes and prokaryotes. In prokaryotes, the translation takes place in the cytoplasm. In eukaryotes, the translation occurs in the cytoplasm or across the membrane of the endoplasmic reticulum. What happens is that when the ribosomes bind to the mRNA, this whole complex then attaches to the endoplasmic reticulum. The new protein is synthesized and released into the endoplasmic reticulum. This protein can be stored inside the endoplasmic reticulum or released in the future, or it can also be secreted immediately. Before going into the details of process of translation, it's important to learn about a few concepts. The main machine which plays a central role in translation is ribosome. Ribosomes are complex molecular structures found inside all living cells, and they act as a site for protein synthesis. The ribosomes essentially consist of two major components, a small subunit and a large subunit. The unit of measurement used to describe the ribosomal subunits is the Swedberg unit, which is a measure of the rate of sedimentation in centrifugation rather than size. In eukaryotic ribosomes, the smaller subunit is 40S and the larger subunit is 60S. When they join, they are denoted as 80S. These subunits lie separately in the cytoplasm until they need to come together for translation. These subunits have different jobs as well. The small subunit reads the mRNA, whereas the large subunit joins the amino acids to form a polypeptide chain.
Okay, class, emphasize ko lang yung two parts, yung large and small subunit natin. Si small subunit natin, class, it reads the mRNA. What do you mean by reads the mRNA? Remember, class, si RNA natin, meron yang nucleotides, di ba? It reads the nucleotides, the codons. Mamaya explain natin ano ba yung codons, no? So basically ito yung ginagawa ng small subunit natin. It reads the content of the mRNA. And then, anong purpose ng large subunit? Ito yung nagpo-produce ng polypeptide chain. Example, sinabi ni mRNA, nabasa ko dito, ang format ng codons is UAAG. No? It will form methionine. So, ang maglalabas ng methionine class, si, tao dito, si large subunit. Tagabasa lang si small subunit natin. So, that is the main goal, no? Main purpose of this two. So, continue natin.
Now, assuming you have watched the videos on structure of nucleic acids and DNA, you know that the information of the genetic code is stored in the form of base pairs with 4 main bases: adenine, guanine, cytosine, and thymine. In RNA, we have uracil in place of thymine. Now, the cornerstone to understand the process of translation is to understand what is a codon. A codon is basically a sequence of 3 DNA or RNA bases. When a mRNA goes inside our ribosome, the ribosome does not read individual bases, but rather these sequences of 3 bases which are known as codons. Each of these sequences of 3 base pairs corresponds to a specific amino acid. For example, if 3 uracil bases are in sequence, which means the code will be UUU, the ribosome will read this code and the amino acid that is used will be phenylalanine. Similarly, if the code is UCC, the amino acid that will be used is serine and so on. The codon AUG codes for the methionine and is also the start codon, which signals the ribosome to start synthesizing the protein. Similarly, we have 3 special codons which signal the ribosome to stop synthesizing the protein. These are UAG, UAA, and UGA. These are known as stop codons, and when the ribosome read these, they know that the protein is now complete and the
So ito yung sinasabi ko kanina regarding codons, no? So we have a long series or sequence of nucleotides. Pero yung codons natin, yung binabasa lang ni small ribosome, no? Small unit, small subunit is by 3, no? Sequence. Either AUG, yan, tulad yan, AUG which is the start codon. And marami pang sequence yan class, no? Ang bahala dyan ay si small subunit. Tandaan natin na pag codon class, it is comprised of only 3 DNA. 3 nucleotides, no? Yun po yun. So, continue natin. So, meron tayong start and stop codon. So, syempre, yung purpose ng stop codon, kapag tingin ng system natin na okay na yung sequence of polypeptide chains or proteins, kinakat na niya yung production. Oh, okay na. Okay na yung haba ng sequence, no? May arginine na, methionine, no? Nakabuo ka na ng protein mo, pwede nang i-stop yung production o yung translation. So, merong start and meron ding stop codon for regulation purposes. Kasi kung walang stop codon, dire-diretso yan, class. Walang regulation. Okay?
their job is done. Now let's get into the details of translation. So the first step in translation is known as initiation. This is a transfer RNA or tRNA, which has an amino acid methionine bound to it. This tRNA attaches to the smaller subunit of ribosome, and when it encounters a mRNA, it starts reading the code on the mRNA from its 5' end. As soon as the tRNA ribosome finds the start signal, the AUG codon, it immediately binds to the larger subunit to form the complete ribosome, and the protein synthesis is initiated. The next step in the process is elongation. Elongation is when the polypeptide chain gets longer. In the larger subunit of ribosome, we have 3 slots or sites which are named as E, P, and A. I'll explain these sites with a simple concept. Like I told you, the first tRNA to attach to this mRNA complex is a tRNA with amino acid methionine. This tRNA occupies the P site. P stands for peptidyl site. Now on each side of the P site, we have 2 more sites, the E and the A site. And here is the mRNA in the smaller subunit. Now what happens is the A site receives the next tRNA which matches the codon next in the sequence. If you zoom in, we can see that the first codon is the start codon, the AUG, to which the tRNA attached will have the amino acid methionine. Now if the next codon is UAU, the tRNA attached to the A site will have amino acid tyrosine. So this tRNA is now occupying the A site. Now we know the main job of translation is to form a protein which is essentially a chain of amino acids. So what we need next is to form a bond between the amino acid on the tRNA in the P site and the amino acid on the tRNA in the A site. After this, the tRNA in the P site loses its amino acid and becomes empty, after which the ribosome moves, and now you can see that the tRNA in the P site moves to the E site, which is the exit site, and the tRNA in the A site moves into the P site. The A site thus becomes empty and ready to receive the next tRNA. So the P site always holds the growing polypeptide chain. This process is repeated again. For example, if the next code is CGU, the tRNA that will enter the A site will have the amino acid arginine attached to it. Next, the bond will form between the small polypeptide chain and the amino acid on the tRNA in the A site. Again, this tRNA becomes empty and the mRNA moves again, repeating the whole process until we get a polypeptide.
Ayan, class. So dito, yung A, no, portion, nagiging blanco siya once na nakapag-produce na ng polypeptide. So, pahaba lang ng pahaba yung polypeptide chain natin, di ba? Nag-start yan with methionine, followed by thymine, and then now arginine. So, pwede pang humaba yan, class, no? Depende kung hanggang saan mag-activate yung stop codon. Pagka na-activate yung stop codon, stop na yung sequence ng polypeptide chain or protein. So, ganun lang yung nangyayari dyan, class.
Chain of desired length. The next step in the process is known as the termination. The ribosomes must have a way to know where to stop so that we get a final protein of desired number of amino acids. This is possible because the mRNA actually contains stop signals, which is known as the stop codon. When the ribosome reads any of the three codons, the UAG, UAA or the UGA, it stops the process of translation and the polypeptide chain is released from the translation complex. So after the ribosome finishes the protein synthesis, is the protein complete? Not really. After that the protein is formed, it goes through some additional processing, like some amino acids may be deleted and some proteins also undergo folding to form a more stable structure. So this was a brief overlook about the process of translation. Guys, thank you so much for watching this video.